Alright, so in this video I'm going to be talking about another approach for event extraction. It is using iteratively improving it by hard expectation maximization. So I'm going to be explaining this paper in 2023, iteratively improving biomedical entity linking, and so we consider these two tasks simultaneously, and we iteratively train them using expectation maximization, giving, for example, fixing one of them as latent variable and then updating the other, and vice versa. Biomedical entity linking and event extraction are two crucial tasks to support text understanding and retrieval in the biomedical domain. These two tasks intrinsically benefit each other, and entity linking disambiguates the biomedical concepts by referring to external knowledge bases, and the domain knowledge further provides additional clues to understand and extract the biological processes, while event extraction identifies a key trigger and entities involved to describe each biological process, which also captures the structural context to better disambiguate the biomedical entities. However, previous research typically solved these two tasks separately. 95% of articles, they do it the same. They do it separately or in a pipeline. But this leads to error propagation. The error propagates downstream, in all of your downstream tasks will be ruined. So it's more challenging to solve these two tasks together. And there is no existing dataset that has both two tasks, so this is another challenge of this paper. To solve these challenges, they propose joint biomedical entity linking and event extraction by regarding the event structures and entity references in knowledge bases as latent variable, and updating the two tasks to specific models in a hard expectation maximization. So we predict the missing variables for each partially annotated dataset based on the current two task-specific models. Update the parameters of each model on the corresponding pseudo-complete task. So it's very a complex idea. I don't think it is it is that much complex inside our brain, because in our brain has a different paradigm. In our brain, it's like microservice architecture, different tasks, different concerns, and they have a very chaotic interaction. So this is an illustration of biomedical entity linking and event extraction, and we use gene ontology to get the definition of the entities. So there is no existing dataset that contains both annotations, for both tasks. And for example, this dataset only contains entity linking, and Genia, is just for event extraction, not entity linking. So that is why it is even hard. So they propose a joint biomedical entity linking and event extraction where each task-specific model incorporates the additional knowledge, the output from another model, to better perform task-specific prediction. To iteratively improve the model specific to each task, we model the entity references in knowledge bases and event structures as latent variables, and devise a hard EM-style learning strategy that has expectation step and maximization step. In expectation, we estimate the missing variables for each partially annotated dataset. In the maximization step, we update the parameters. So they propose a joint biomedical entity linking and event extraction. so two models can mutually improve each other. They design a collaborative training strategy because it's iteratively optimizing the two tasks. And our joint framework consistently achieves significant performance gain for each individual task. So we have biomedical entity linking. Given a text and a set of spans, we have N indicates the number of tokens, P indicates the number of mentions, and we have biomedical event extraction. Biomedical event extraction consists of two subtasks: event detection and argument extraction. Illustration for our joint for EEE for biomedical event extraction. So it induces activation of signaling with production. As you see, we are using Go, this ontology. So we present our joint event extraction and entity linking framework consists of an entity-aware event extraction module that leverages the additional knowledge from knowledge bases such as Go, UMLS, and an event-aware entity linking module which utilizes event structures to characterize the biological processes that each entity mention is involved and disambiguate its meaningful so that we can better link each entity mentioned to the correct reference. The base event extraction model takes a text and the set of all entity mentions in X. We first encode X with a PLM, pre-trained language model, to obtain the contextual representations for the text. So for each token J, we feed its representation into an event type classification. For argument extraction, we concatenate the contextual representation of each identified event trigger with the representation of each argument candidate. So we propose a dual encoder framework that incorporates the external domain knowledge of the given entity by the base entity linking model. When we take more than 30 candidates, the fraction doesn't increase. And we apply base entity linking to select a biomedical concept from candidate set of concepts retrieved from the Go knowledge base. And then we obtain the definition of those biomedical concepts Go and use it for this model. So we apply additional pre-trained language model-based knowledge encoder that specifically takes in the selected biomedical definitions for M sub I and encodes it in contextual representation. We take the contextual representation of CLS token as knowledge encoding for M sub I, and we adopt the same process for all entities. We obtain a knowledge enhanced entity representation. So this is the knowledge enhanced representation, and then we concatenate the representation of each identified event trigger with the enhanced entity representation. This is the enhanced. And then feed it to the model to get the probability. So we have base entity linking model for each candidate C sub K. We obtain its definition in Go. So we have the definition and sentence, and then we can predict the probability. So given an input text, first we apply base event extraction to obtain the pseudo trigger and argument role labels. Second, we take one entity mention and retrieve candidate concepts. We inject event information into input sequence of the entity linking model. Each entity only participates in a single event with a unique argument role. For the event extraction task, we denote the training instance where x sub I is a sentence and y sub I is an event annotation. So we prepare the candidate biomedical concept for both entity linking and event extraction. And we randomly initialize the parameters for joint for event extraction and entity linking. So to first obtain a initialized base model, we individually train on the label entity linking and train event extraction on the labeled entity linking until the model converges. So in the expectation step at the beginning, we first initialize 2 empty sets for collecting pseudo-label instances. We run the entity linking model on the event extraction dataset to generate pseudo-entity linking annotation. For each instance in the event extraction dataset, we run the entity linking model and predict pseudo-entity linking labels. And in the maximization step, for the event extraction, we loop through examples in the newly collected event extraction dataset. And this f sub theta optimizes the log likelihood of the true event extraction label based on the discrete latent variable. So these are example sentences and results for event extraction at each round sampled from Genia 2011 development set. We have text, we have previous results, we have entity knowledge from joint entity linking, and then now you see that you can get the trigger much easier, the theme and the cause for each trigger. Another set of sentences. So this is the algorithm. First, we get the entity linking dataset, also event extraction dataset, 2 datasets. Also knowledge base, because that Go, do you remember, gene ontology? And so after we get all of them, we retrieve the set of candidates for mentions. And then we initialize the entity linking and the event extraction model. And then we go through these are optimization step: expectation, maximization. So these are methods. You see that it's still we have a very low accuracy in the world because state of the art is around 64. So strange. And for recall is 59, for precision is 69. But this is important. You know, a fun score is just 64. It's just less than 70%.